NASA has released the results of its twin study. 10 research teams worked for this and published the combined summary paper in the Science magazine. The NASA twin study was a 340-day investigation conducted by NASA's Human Research Program (HRP). It studied identical twin astronauts, Scott Kelly and Mark Kelly, while Scott was in space and Mark was on Earth. Scientists conducted a year-long biomedical study of their health. The twin study offered a unique opportunity for NASA to better understand how long-term spaceflight affects a human body. Because identical twins share the same genetic makeup, they are physically very similar. Even after Scott landed back on Earth, the researchers of the twin study continued to gather data and analyze the results from their biomedical investigations. NASA scientists compared genetic, physiologic, and behavioral changes in the identical twins Scott and Mark Kelly before, during, and after Scott embarked on a one-year mission on the International Space Station from 2015 to 16. According to this study result, some biological functions were not significantly affected by spaceflight. Long-term spaceflight causes more changes to gene expression than shorter trips, especially to the immune system and DNA repair systems. Scott experienced thickening of the carotid artery, thickening of the retina, weight loss, shifts in gut microbes, reductions in cognitive abilities, DNA damage and changes in gene expression, and a lengthening of the ends of chromosomes called telomeres during the flight. Upon return to Earth, the telomere elongation was replaced by accelerated shortening and loss, a potentially negative consequence for cellular health. The study will help scientists better understand the changes astronauts undergo during long-term space flight. It may also help them better treat or prevent any damage to astronauts' health caused by prolonged space flight, which is especially important as NASA and other groups prepare for 3-year missions to Mars. Given that the majority of the biological and human health variables remained stable or returned to baseline after a 340-day space mission, these data suggest that human health can be mostly sustained over this duration of spaceflight. To date, 559 humans have been flown into space, but long duration, that is more than 300 days missions are rare. Long duration missions that will take humans to Mars and beyond are planned by public and private entities for the 2020s and 30s. Therefore, comprehensive studies are needed now to assess the impact of long-duration spaceflight on the human body, brain, and overall physiology. The space environment is made harsh and challenging by multiple factors, including confinement, isolation, and exposure to environmental stresses such as microgravity, radiation, and noise. Key results from the NASA twin study include findings related to gene expression changes, immune system response, and telomere dynamics. Other changes noted in the integrated paper include broken chromosomes rearranging themselves in chromosomal inversions and a change in cognitive function. Many of the findings are consistent with data collected in previous studies and other research in progress. The telomeres in Scott's white blood cells, which are biomarkers of aging at the end of chromosomes, were unexpectedly longer in space than shorter after his return to Earth, with the average telomere length returning to normal 6 months later. In contrast, his brother's telomeres remained stable throughout the entire period. Because telomeres are important for cellular genomic stability, additional studies on telomere dynamics are planned for future one-year missions to see whether results are repeatable for long-duration missions. A second key finding is that Scott's immune system responded appropriately in space. For example, the flu vaccine administered in space worked exactly as it does on Earth. A third significant finding is the variability in gene expression. While in space, researchers observed changes in the expression of Scott's genes, with the majority returning to normal after 6 months on Earth. However, a small percentage of genes related to the immune system and DNA repair did not return to baseline after his return to Earth.